Welcome to chapter 16, all about DNA. So, first of all, DNA is made of nucleic acids, hence the name deoxyribose nucleic acid, and really it's made up of a whole bunch of nucleotides. You should remember this from chapter 5. So, in every nucleotide, there's a phosphate, a sugar. The two types of sugar are deoxyribose and DNA, and ribose and RNA. Sorry, you can't see all of that. And then nitrogen base. And those are really the main part we talk about. And so most of the time I'll refer to them as A, G, C, T, and U, which is only found in RNA. So here's a picture of each of the different nitrogen bases. DNA does not have any uracil. RNA doesn't have thymine, so those are used interchangeably depending on if you have DNA or RNA. Purines, which are A and G, they have 2 rings. Pyrimidines, T, C, and U, have 1 ring. And so, as I talk about DNA and DNA replication, the carbons are going to become really important. We're constantly going to be talking about 5' and 3', because some things will attach over here, some things will attach over here. So those 5' and 3' carbons. So the backbone, you're going to have the phosphates and sugars, and the bases are going to be in the middle. And so the phosphate is always on the 5' side, and then the 3' side over here is phosphate, but that's where the next nucleotide attaches. So here shows the entire piece, so kind of cartoonish, a little bit more like it's a space filling, and the actual what the chemical structure looks like. So you have your phosphate and sugar backbone, your nitrogen bases here in the middle. They do run anti-parallel, so you have their 5' to 3'. The other side, you're going to get the 3' to 5'. And so it's really like DNA is upside down. So as far as bonding, there's hydrogen bonds down the middle and covalent bonds down the sides. And those hydrogen bonds, they're considered weak bonds, but a lot of them could be strong. And so this helps with replication, because when it replicates, it's going to go right in the middle. So those bonds are easier to break than these ones on the side. Base pairing, so you have your purines again, which have the 2 rings, and pyrimidines that only have your purines have 2 rings. This way, pyrimidines have 1, and they're always going to pair together. So A and T always pair together. There's 2 bonds between them. C and G, there's 3 bonds between them. Chargaff is the one who found this. He found that in organisms that A was always relatively equal to T, and G was always relatively equal to C. And also, that makes a purine always bonding to a pyrimidine. Otherwise, it just wouldn't fit. And as far as the way DNA replicate, we have found it is the semi-conservative. These other 2 just were not actually found to work. Just kind of bringing in prokaryotes, bacteria, because we will be talking about them later. The biggest difference with them is that they're in a circular loop instead of a double helix. And so they have a place where they start and they keep going around. I'll show those videos in class, so and I'll attach a link to just the PowerPoint, so if you want to go back to those videos you can. So how does DNA actually replicate? There's base pairing, as we talked about, A and T, C and G, and so that serves as a template for the next one. So here's A, the new nucleotide knows that it has to be a T. So each new strand is half the old parent and half the new. And that's the semi-conservative. So first step, we have to be able to unwind DNA. The enzyme that does this is called helicase. So it unwinds part of the DNA. You can see here and here. and this is stabilized by different binding proteins. What you can't see there is this is called the replication fork. So next step, so now we're unwound. Now we have to build that new DNA strand. And so we have DNA polymerase. And number 3 is our first one. And it's going to come and help us build that new one. So what about energy? It would take energy to do this, right? So if you look, this molecule might look familiar. An A and 3 phosphates, ATP. So it's really just a nucleotide. And so when we take that phosphate off, it's ADP. And we get energy. So there's other ways, right? Of course. I can go down to 2 and I can get AMP. Not as easy, but it comes with its own energy. And they leave behind a nucleotide. You can also get different types, GTP, TTP, and CTP. So one of the first things that happens, nucleotides arrive in nucleoside triphosphates. DNA pairs with that triphosphate, gets that energy for bonding. And so they already have their own energy. They're bonded by the enzyme DNA polymerase. And they can add. So we can only add nucleotides from the 3' end of the growing DNA strand. So they need a starter that grows from 5' to 3'. There's the energy. Notice those phosphates coming off. The energy. Phosphates have to come off. Energy. Phosphate coming off. Energy. 5' to 3'. So that the strand is only going to grow 5' to 3'. Here's our completed piece. So taking it apart, it's helicase, showing it happen at both sides, 5' to 3'. So we need a primer to add on to. So those, and it works. Now on this side, there's no energy to bond because it's already taken off. So if you go back, up there, there's no energy. So that doesn't work. We can go back and basically does it backwards. It has a 3' to 5'. And so ligase has to come through and bond those last ones. So you have leading and lagging strands. There's our helicase and our binding proteins. The replication fork is going this way. You have DNA polymerase, who's able to go 5' to 3'. Everything works out handy dandy. Yay, we can replicate easily. But this way, it doesn't work. So it has to go out this way. So it can only build from the 3' end. And these are called Ozaki fragments. And ligase has to come, and it's like glue to glue these together. So you have your leading strand and your lagging strand. The leading strand has continuous synthesis. The lagging strand has to be joined by ligase. So you get your bubble here. Polymerase works in one way, and so it's not like it's always on top leading strand, whichever way is 5' to 3' for it. And then going back the same way, so you can see it switches based on where that came. So to start this whole thing off, we need RNA primers. You get something called primase, and it's gonna help get to that DNA polymerase. So that primase is that little red piece over here. And it's a starter sequence. Let's see. Those eventually go away. So DNA polymerase 1 comes through, removes those sections of RNA, and replaces them with DNA nucleotides. But polymerase 1 can still only go onto the 3' end. There's some RNA there. All the DNA polymerases slowly go there. So that part gets in, and we have an issue. So there's a loss of bases at the 5' end in every replication. Your chromosomes get shorter each time you replicate. So is there a limit to how many cell divisions you can get? We have found that there's things called telomeres. And so the primer can be removed, but it's not replaced because there's nowhere for it to go. So telomeres are repetitive sequences added to the things. So the shortening of telomeres can play a role in aging. So remember A's, those are always enzymes. So that enzyme will add telomere segments in cells that have to go divide frequently, skin cells, stuff like that. Cells with increased telomerase activity allow them to keep dividing. So this way we have our binding proteins, we have our helicase opening everything up, we have our primase mostly made up of RNA, we have DNA polymerase that can easily bind here. We have our Saki fragments because they're basically binding the wrong way. DNA polymerase 1, the leading strand and lagging strands. Going into the DNA polymerases again. Polymerase 3 can get 1,000 bases per second. So this is our main builder, whereas polymerase 1, 20 bases per second. It's not a very fast process. It's good for editing, repair, and that primer removal. 2 found them. So E. coli can take about 1 hour to copy. That's 5 million base pairs in a single chromosome. Divides, has 2 identical cells. Human cells copy its 6 billion bases and divide in only a few hours. So, pretty good. And there's only about 1 hour error per every 100 million, 30 years per cell cycle. Not bad. So, how do they do it? They can get a lot of typos, but polymerase 1 will actually come through and proofread. So, one thing is like a bulge, so if you have 2 purines together, so they can repair mismatched bases, abnormal bases, damages throughout the life, and we get error rate that is reduced from 1 in 10,000 to 1 in 100 million. So that polymerase is working pretty good. There are some that are not caught, so carcinogens, especially these different pieces of cutting errors, and I have to be done.